When some people think about science, all they think about is a body of facts. But science isn't just a body of facts. It's a process. A process where we can try to find an explanation for something we don't know. I like that because it tells me that the universe is predictable and orderly. If the universe wasn't predictable and orderly, then science wouldn't be possible. There would be no good way to find an explanation for something if it were random and chaotic. But because the universe makes sense, we can explore it, and we can use science as a tool, a process to help us discover things about the universe. Good science starts by making good observations. Observations can be quantitative, meaning you're going to take a measurement of something. Like, I'm going to measure the length of this pencil. Or maybe the volume of this bag. That's a good observation. Qualitative observations have to do with things like color changes and the quality of things. Like that it's clear. But if I said the bag was half full of water, that would be which observation? Quantitative or qualitative? Well, if I say the bag is half full of water, that's a quantitative observation because we're using the number half full of water. What if I'm wondering about the length of a long femur bone, the long bone in your leg? How does the length of this bone compare to the total height of an individual? I'm not sure. That's a good observation, that these bones have length, and that there might be a proportion between the length of a long bone and the height of an individual, the total height. That's an observation. Now, one thing I've noticed is that when I use this bromethyl blue in these clear solutions, some of them change color. It's an observation that I've made. And I want to use the process of science to figure out what's going on. And if I add some bromethyl blue to the first one, it stays blue. If I add it to the second one, it turns yellow. So the question is, why does the same solution turn one colorless solution blue and the other colorless solution yellow? So here's my data. I've written the chemical formula for all of the solutions. And I have two that were unknown. Now the first one I knew stayed blue. And then when I put it into when I put the bromethyl blue into the hydrochloric acid, it turned yellow. So I came up with a hypothesis. If bromethyl blue is an acid-base indicator, then acids will turn yellow, bases will stay blue. So I look to see which are acids and which are bases. That's going to be based on the chemical formula. Chemical formula for acids have the hydrogen in the front. The chemical formula for the base has the OH in the end. So if it starts with a hydrogen, it should turn yellow. If it ends with the OH, it should stay blue. So KOH, that should stay blue. HNO3, that should turn yellow. CaOH should stay blue. So if they're a base, they stay blue. If they're an acid, they turn yellow. So if this one stayed blue, if I check unknown number 2 and it turns yellow, then I know that it's an acid. What about this down here? ZnNO3. Well, that's neither an acid nor a base. So would I expect it to turn blue or yellow? Well, neither. I would expect it to turn something other than blue or yellow, which it does. In this case, it turns green. What's going on here? So I make a hypothesis. And then based on that hypothesis, I try to collect some data, I make some more observations, and then I can perform some more experiments. So first I'm going to make some observations. What are these solutions, and what's their chemical formula? All right, now I'm going to test my hypothesis. Does the solutions that are acidic turn yellow? And are the ones that are basic turn blue? So I'm going to put a drop in each of these wells, and now I'm going to see if I'm correct. If the ones that are acidic are yellow and the ones that are basic, if they turn blue. So if this is an unknown, I don't know what that one is. If it turns yellow, I can have a pretty good idea whether or not it's an acid or a base. This one turned green, which tells me it's neither an acid nor a base. Will this process work all the time? Coming up with an experiment based on some observations, recording the data, making a conclusion based on the experiment. Is that the only way that science works? The answer is no. Some experiments or some questions are just too big. There's no way to really have a controlled experiment to figure it out. We have to use other methods. One of the times when we have to use another method is whenever we're testing things like drugs and pharmaceutical products. Those experiments are totally different. There are two different ways of thinking scientifically. One is inductive reasoning. In this case, you accumulate facts, and then based on the facts, you come up with a conclusion. So for example, I might see some tracks in the room, I might see a broken lock, I might see some people that have recently left the room, and then based on those facts, I come up with the conclusion, Joey stole something from the drawer. Now, that's inductive reasoning. I accumulate the facts first, and then based on the facts, come up with a conclusion. Deductive reasoning is different. Deductive reasoning, I start with a conclusion, and then I see if the facts support that conclusion. Now, normally this is a scientific method. We start with our conclusion, our hypothesis, and then we see if the facts will support that hypothesis. Does that process work for everything? The answer is no. There are some things where the scientific method, as I described it, doesn't work. And that's because there are bigger issues and it's hard to get a controlled experiment where there's only two variables, where there's only one thing or another, an acid or a base, and where there's nothing else that's influencing the experiment. Now, a good experimental design will try to eliminate as many of those other variables as possible, but that's not always a possibility. For example, when you're testing pharmaceutical products, so there's a lot of things that go into it. The health of the individuals, the age of the individual, maybe even the gender of the individual, their lifestyle habits, their genetics. There's a lot that goes into it, but we do our best to get a sample of the population that we're going to test with our new pharmaceutical product. The other group is going to get a placebo, a sugar pill. Now, the problem is that there can be psychological effects if the group thinks that they're getting the actual medication. So what can I do to eliminate that problem? Well, I can make it so that the group that gets the pill and the group that gets the placebo, they don't know what they're actually taking. That this group doesn't know if they're actually taking the pill, and that this group doesn't know that they're actually getting the placebo. But there's another problem with that. I know. If I know that this group is actually getting the pill, and this is actually getting the placebo, and I really want my pill to be this great pharmaceutical product so I can make lots of money, that's going to influence what I see in the results. So I need to figure out a way where one group can get the pill, the other group can get the placebo, but the people in the group don't know what they're actually getting. But I can't know who's getting the pill and who's getting the placebo. We call that kind of a study a double-blind study. I am blind. I don't know who gets what. And the Subjects are blind. They don't know who gets what. So we're both blind to try to eliminate some of that bias and to eliminate some of the craziness of the results. Another time when the scientific method, as I described it, with only 2 variables is nearly impossible, is when you're doing an epidemiological study. That is, you're trying to find a pattern in the population. So who's more likely to have a heart attack? White Caucasians or African Americans? Well, there's, again, lots of variables. Diet, exercise, age, all these things go into it. But if we look at a large cross-section of the population, we can try to eliminate as many variables as possible and just see what the pattern is in the population. Who's more likely to get diabetes? People born before 1960 or people born after 2000? Does that mean I believe every scientific study I hear? And the answer is no. Because remember, a good experimental design is important. Otherwise, there's always other variables, and the data can be twisted to make it seem like one thing or another. Or the experimenter introduces bias into their experiment. So use some critical thinking. Don't just accept it because it said it. Does it make sense? Has it been verified? Is it repeatable? Has it been evaluated? Does it stand the test of time? So use some critical thinking, ask some good questions. Because ignorance is dangerous. There's lots of people out there that just believe anything and everything they hear. So they see this scientific study about how you take this pill or this powder, and you can go from this little wimpy guy to this really muscular one. But is there more to it than that? Is it that the muscular guy was also exercising regularly? Is it just based on taking the powder? Or do you also need to exercise? Is it the kind of exercise that matters? Maybe this guy, maybe the guy wearing the glasses is just doing aerobic exercise where the muscular guy is lifting weights. So we want to know and ask some questions whenever we see a study. Is it the results consistent and reliable? Like, was the study done in a consistent, reliable kind of way? Were the results interpreted correctly, or did they twist the data to say what they wanted it to say? Was there a source of bias? Was it just the company who's producing the weightlifting powder the one doing the study? Because, of course, they want to see it be successful. And are there any other possible explanations? Is there something else that might have happened to cause those results?